Hi everyone, you are watching Biology with Inshara, and in today's video we are going to talk about osmosis. Now this is the part 2 of the video, and if you have not seen part 1 of the video, then I highly recommend that you stop watching right now and go and watch part 1 of the video first. You can find the link in the description box down below, so just click on the link and you will be directed to the video. Now you need to understand that if you have not seen part 1 of the video, then you will not understand this topic, as there are certain things that you need to first understand in part 1, because that is the basics or the beginning of the chapter, and we're going to complete osmosis in this video, and then in the next video, which I will be making next week, we're going to talk about active transport, which is very small and it shouldn't take too much time because it's easy. Okay, let's get started. Right, so in this topic, in this video here, we're going to study what happens when you place a plant cell in different osmotic conditions, and the three main osmotic conditions are isotonic, hypotonic, and hypertonic. Okay, so let's first discuss isotonic. What is an isotonic osmotic condition? And now this means when the water concentration is the same in both the cell and the surrounding solution. So just to clarify that, and just to further explain it, I've written down that water potential is the same in both the plant and solution. So what happens is that there is no net change in the cell. It remains the same. The cell itself remains the same. It doesn't get big or doesn't get small. It remains the same in its original regular shape. Water moves both in and out to maintain that favorable condition. Now in part 1 we discussed that certain things need to get in the cell for survival, like sugar, water, glucose, and then there are waste products which could involve water. Water is also a waste product, and that then water can also leave the cell along with a few other materials such as carbon dioxide and more. And when materials move in and then they move out, now this ensures that the cell doesn't get too big or it doesn't get too small, because then again the moving in and out of materials maintains that state of equilibrium, resulting in an isotonic solution state, which means that this is the most favorable condition of any cell, including the cells in our body and including for plants, because this is their original shape. Okay, hypertonic. Now what happens in hypertonic is that water potential is higher outside of the cell. Now the water concentration is higher outside of the cell. So you can imagine that from the definition of osmosis, water should move from the solution into the cell because the concentration is lower in the cell. So when water moves into the cell, what happens is that more water collects in the vacuole, which you can see is blue here, right? So when more water collects in the vacuole gets bigger due to the increase in volume. So when the vacuole gets bigger, it puts pressure on the cytoplasm, and then the cytoplasm also gets bigger. And when it gets bigger due to the increase in volume, it pushes against the cell membrane, and then the cell membrane pushes against the cell wall, and this creates an internal pressure. So let's read this. Vacuole becomes turgid. This is what it exactly means, that the vacuole gets bigger and then the cytoplasm also increases in size, and then both the vacuole and cytoplasm push against the cell membrane, and then the cell membrane in turn pushes against the cell wall, and this here creates an internal pressure. Now if you can imagine, if this internal pressure, if it hadn't been for the cell wall, then the cell would have exploded, such as in the case of an animal cell. Since we don't have a cell wall, the cells in our bodies would explode, right? Our cells don't have a cell wall. But in the case of a plant cell, the cell wall is made up of a fibrous, strong, rigid structure, which is a polysaccharide, a complex sugar called cellulose. Now, cellulose cell wall is very, very rigid, hard, and it is able to withstand the pressure within the cell, that internal pressure. Now, this black dotted line here, not dotted, well, straight line, this is actually the cell wall, the outermost or membrane of the cell. Now the cell membrane is too thin and delicate, so it will not be able to withstand the pressure. Think of it as like this. Now take a rubber band. A rubber band is elastic, so when you stretch it or pull it, it eventually breaks. But with the case of a cell wall, a rigid cellulose cell wall, it is inelastic, and no matter how hard you stretch, push it, no matter how much strength you use it to push or stretch it, what happens is that the cell wall does not break at all, because it is very, very hard and strong and it is impossible to tear it, because then again, just like I said, it is a very strong material, a very strong fibrous material. Okay, let me just get my pointer and let us move on to hypertonic, which you can see is here. Now this is slightly more complex, so bear with me and I will get you through in a couple of more minutes. Now in hypertonic, the water potential is lower outside. So you can imagine when the water potential or the concentration is lower outside, that basically implies that it should be higher within the cell. And with that being said, water moves out of the cell. You can see that water is moving out in the picture here. Okay, now I just forgot to mention, let me just mention this here again. When water moves into a cell, it becomes turgid. Okay, this is called turgid, and this is also normal for a plant cell because this helps to support neighboring cells, and this is also good for the plant because then again they're having this state of protection due to the cell wall, and then one turgid cell supports another neighboring cell. So this is considered good for a plant cell. Okay, right, just to continue. So when water moves out of the cell, just like here, it is moving out. No, you can. It's very logical to think that the volume within the cell will decrease. So the cytoplasm and vacuole are using water because most of the water can be found in the cytoplasm and vacuole. So they shrink. They become smaller. Cytoplasm shrinks, and also the vacuole shrinks. And when they all get smaller, the cell membrane pulls away from the cell wall. Now let me explain this in more detail. Now this here in red is the cell wall, okay, and now this here in black is the cell membrane. Now you may be thinking that, to be very more precise, normally in the case of a normal cell, this here is the cell wall, and then this, the innermost membrane, is the cell membrane, and there's this small space in between. But in the case here, you can see that just hold on one second. Yeah, so in the case here, this is the cell wall and this is the cell membrane, and when this is the vacuole, the blue region is the vacuole, and then the cytoplasm is the brown region. So when they both shrink, the okay, so when they both shrink, what happens is that the cell membrane pulls away, which you can see here, it pulled away from the cell wall. Now you can see that this region that I'm going to shade right now, color for you, these regions are the spaces that I'm going to further discuss here. Now this space has been created between the cell wall and the cell membrane. Okay, this internal space has been created between the cell wall and the cell membrane. So what happens is that the cell membrane here, where was I? Yeah, so the cell membrane pulls away from the cell wall, creating a space in between the cell wall and the cell membrane. And when this happens, the cell is in a state of plasmolysis, or it is said to be plasmolysed. Plasmolysis or plasmolysed means when the plant cell has lost too much water, due to which the vacuole in the cytoplasm shrink or get smaller, due to which the cell membrane pulls away from the cell wall, leaving this internal space between the cell wall and the cell membrane. Now what happens is that, this is not the end of the story, although it is losing water, but what happens is that eventually the cell will lose all of its water outside, and then it will reach a point when the concentration of water will get higher outside of the cell, right? I'm coloring this blue so that you can understand. It will get higher outside of the cell in the solution. So what will happen is that water will move back in via osmosis. It moves back in, and when it moves back in, it will actually fill up the spaces between the cell membrane and the cell wall. Now I'm coloring this with black because then that water will then move in or diffuse in between the space between the cell wall and the cell membrane. So then water will be covered here. It will be filled. This is what happens in the case of a hypertonic solution. This is slightly more complex, but nothing that difficult. Now, thanks for watching, everyone. This is it. There's nothing more to this. This is the end of the topic. Just watch part 1 and part 2 and you will get a better idea of this video. Any questions, any requests, then let me know in the comment section down below. And if you're watching this on Facebook, then like and follow my page, share the video and the link to the Facebook page with your friends and families. And if you're watching this on YouTube, then do subscribe and like. Leave a comment down below, and don't forget to share this as well. And lastly, whether you're watching this on Twitter or Facebook or YouTube, keep learning, because I make weekly content for you guys. As always, see you in the next video.